Welcome, AP Biology students. This is our lecture on meiosis and the sexual life cycles. Ultimately, what you must know from this unit, you should be able to distinguish asexual reproduction from sexual reproduction, be able to discuss the role of meiosis and fertilization in sexually reproducing organisms, understand the importance of homologous chromosomes as it relates to meiosis, know how the chromosome number is reduced from a diploid number to a haploid through the stages of meiosis. Understand three important differences between mitosis and meiosis, and also know the importance of crossing over, independent assortment, and random fertilization as it relates to increasing the genetic variability of an organism or population. Genes are segments of DNA that code for basic units of heredity. Offspring acquire genes from parents by inheriting chromosomes. So remember that genes are those functional units of DNA that will code for a specific trait. And we'll look at this a little bit more in genetics. Offspring ultimately acquire those genes from the parents by inheriting those chromosomes through reproduction. So if we look, here is a picture of a family. And those two types of reproduction are both asexual and sexual reproduction. Asexual reproduction is going to produce clones that are genetically identical to the parent. Often, what those clones are female, and they look like the parent themselves, so it will be looking like the maternal organism. Meiosis is the process that's going to produce gametes. Remember, gametes are the sex cells. So meiosis produces gametes, unlike mitosis that produced new somatic cells or body cells. Asexual reproduction involves a single parent. Sexual reproduction involves two parents, male and female, where you're going to have the exchange of gametes, sperm and egg. In asexual reproduction, there's a little variation in a population, and any variation that does take place is only through mutations. In sexual reproduction, there is lots of variation within a population, and that ultimately will lead to diversity among the population. Asexual reproduction, it's an advantage because it's fast and energy efficient. Sexual reproduction, on the other hand, is slow and energy consumes a lot of energy. Ultimately, for generation to generation, sexual reproduction is the more advantageous type of reproduction in that it does increase the genetic variability of a population. Examples of asexual reproduction include budding or binary fission. Examples of sexual reproduction would be things that we see in humans. Certain trees are all carrying out sexual reproduction. So here you can see redwood trees as an example of your sexual reproduction, and a hydra. This is the parent hydra, and then this little bud here. This is an example of budding. It just grows like a little mini-me off the side there. So let's take a further look at chromosomes. Ultimately, we know that chromosomes are found in our cells, and the human body, or somatic cells, their diploid number, 2n, is equal to 46 chromosomes. Each pair of homologous chromosomes includes one chromosome from each parent. Remember that homologous chromosomes are chromosomes that are similar in the size, in the shape, and the genes that they carry. The first 22 pairs of chromosomes do not determine sex. We call those the autosomal or autosomes. The autosomal chromosomes, labeled pairs 1 through 22, are different from that of the sex chromosomes, which is pair 23. The sex chromosomes are X and Y. The female, for humans, female sex chromosomes includes XX. The male sex chromosomes are the X chromosome and the Y chromosome. So if we look at it, gametes themselves, n equals 23, includes 22 autosomes plus one sex chromosome. So an example will be 22 plus X. If you look at a sperm, it would be 22 plus X or 22 plus Y. So the female egg always carries the X sex chromosome. It's the male sperm that will carry either the X sex chromosome or the Y sex chromosome, ultimately determining the gender of the child. Homologous chromosomes in a somatic cell. So if we look at the homologous chromosomes, if we're looking at 2n equals 6, in red we have the maternal set of chromosomes, n equals 3, and we have the paternal set of chromosomes, n equals 3 in blue for the parent, father. So we have sister chromatids is equal to 1 duplicated chromosomes, and then together we have 2 non-sister chromatids in a homologous pair. Pair of homologous chromosomes, 1 from each set. Remember that those sister chromatids are attached to each other at their centromere. If you look at a picture of the chromosomes lined out according to their homologous pairs, that is what we call a karyotype. And a karyotype is a picture of an organism's complete set of chromosomes that are arranged from largest to smallest pair. At the end there you can see autosomal pair 22, and then of course you have the sex chromosomes, X and Y. So if you look, here is a pair of homologous chromosomes. You can see where the centromere is located. You can see sister chromatids. Over here you can see metaphase chromosome. So ultimately you could see that these chromosomes themselves are similar in size. They are similar in the shape, especially with where their centromeres are located. And those color patterns represent different genes that they carry. So they have the same genes. Making a karyotype, unsorted chromosomes, basically you'd have to sort these and line them up from largest to smallest. And if we were to look at this, looking at this karyotype right here, would you determine this one to be male or female? If you answered female, you are correct. If you take a look at this karyotype, chromosomal squash, would you consider this one to be a male or a female? If you answered male, you are correct, because we have the X chromosome and the Y chromosome present. So karyotype is used to determine genetic abnormalities. And if you look, you could see in this karyotype what the chromosomes would all look like. Now let's look at some abnormalities. Cancer cells, some have abnormal numbers of chromosomes. So if you look at each pair, you get an abnormal amount of chromosomes at each spot. So the karyotype of a metastatic melanoma will look something like this. Here's a karyotype of breast cancer cells. All right, the next concept deals with these things called HeLa cells. And HeLa cells are the oldest and most commonly used cell line in biology. Basically, HeLa cells is derived from the lady from which these cells were harvested from by the name of Henrietta Lacks. Basically, what happened there is she had cervical cancer, and cervical cancer cells were taken from her back in 1951. Her doctor took those cells without her knowing, without her giving any acknowledgement or permission to do so. So what are these HeLa cells? HeLa cells, or immortal cells, do not die after a few divisions. They have active versions of telomerase, and when looking at, we'll look at that a little bit later. But ultimately, these HeLa cells are used in research. They've been used to develop a vaccine for polio. They've been used in research for cancer, AIDS, looking at research for certain viruses. They also play a role in radiation research. It is estimated that the cells produced in culture exceeded the number of cells in Henrietta's body. So ultimately, if you look at a HeLa cell karyotype, here's a picture of what one would look like. So looking a little bit more, as I trek through time, HeLa cells have aided in scientific advancement, but at what cost? Well, in 1951, there was a biopsy. So here you can see Henrietta. The tissue was taken from Henrietta Lacks without her knowledge. In 1953, that's when we developed the polio vaccine. Jonas Salk was supplied with HeLa cells from a cell culture factory established at a university. And then less than a year later, Salk's polio vaccine was ready for human trials. Cloning in 1955. Basically, we saw some cloning at the University of Colorado in Denver, successfully cloned the first human cells. 1964, we got into ethical issues where scientists injected HeLa cells into test subjects to show how cancer basically could establish in the medical, looking at internal medical review boards, so on and so forth. 1969, HeLa cells were used to study the human papillomavirus. Then we had another ethical concern in 2013, where it just keeps on going through this huge kind of history of what they're used for and why it became an ethical issue. There is a good book out. The book is called The Immortal Life of Henrietta Lacks, that you could buy and read all about her story and HeLa cells. So let's take a closer look. Here it is. So controversy was that the cells were harvested without the patient's consent. Discarded tissue can be commercialized and sold for profit by companies. And the genome, which was published in 2013, the genome of these cells was published without the family's consent itself. So there you can see a picture, and here you can see a picture of the family and Henry Lacks in August 1st, 1920, and then October 4th, 1951. So the life cycle, reproductive history of organisms from conception through the production of its own offspring. Basically, you have fertilization and meiosis that are going to alternate in sexual life cycles. In meiosis is cell division that is going to reduce the number of chromosomes. 2n equals n is going to create those gametes. In mitosis, the daughter cells are all going to be diploid in nature. So you ensure that each new cell gets a copy of the DNA. So each new somatic cell. But in meiosis, you want to reduce that number. And that's why we go through two successive divisions. So ultimately then we reduce that number, so then upon fertilization, when the gametes are going to combine, the sperm and egg, you are going to restore those those haploid numbers back to their diploid number. So the sperm for a human would have 23 chromosomes, the sperm or the egg for a human would also have 23 chromosomes, and when combined you'd restore that 46 diploid number. So fertilized equals the zygote, and that would then become 2n. The zygote then will undergo division by mitosis to make a multicellular diploid organism, and that's how we would grow, and any multicellular organism would grow. So we do see varieties of sexual life cycles. There you can see what a life cycle will look like in an animal. Here are some plants and some algae. Here are most fungi and some protists. So you can see you have the adult form, diploid multicellular organism, undergoes meiosis to produce the gametes. So the gametes are N. When two gametes would meet then, upon fertilization, you restore a zygote, which is diploid in nature. Many plants and some algae, basically you have a diploid multicellular organism or sporophyte. The sporophyte is going to release spores or pollen or something of that sort to then be haploid in nature. Ultimately, then mitosis would occur. You have a haploid multicellular organism that would be known as the gametophyte. So the diploid multicellular organism is a sporophyte. The haploid multicellular organism is known as your gametophyte. And ultimately then when the two haploid cells would meet upon fertilization, you then restore that zygote, which is diploid in nature. Most fungi and some protists, you have a zygote. Meiosis will form two diploid or two haploid cells. Mitosis would occur, and you would have that organism again. Haploid unicellular or multicellular organisms, and then upon fertilization, you would come back to that diploid zygote number there. In the human life cycle, you have a multicellular diploid adult, so 2n equals 46 chromosomes. And here you can see in the gonads, you have the testes for the male, you have ovaries for the female, which are going to produce the sex cells. So the sex cells are going to undergo, the gonads will undergo meiotic cell division to produce the gametes. So in the ovary, you're going to produce the ovum, which is the mature egg. In the testis, you're going to produce a haploid spermatozoa, which will later mature in the epididymis, which is the series of tubules that are on top of the testis. And upon fertilization then, when the sperm and the egg meet, you are going to restore that diploid number of 46 in the resulting zygote. And then from there on, that zygote will undergo mitosis, and development will take place to produce the adult human organism. So let's look at meiosis. Meiosis is equal to a reduction division. Ultimately, we're going to have three things going on here in the life cycle of the cell or the formation of the cell. We'll have interphase, then we'll have meiosis 1, and then we'll have meiosis 2 take place. So we have a pair of homologous chromosomes in a diploid parent. The chromosomes are going to duplicate. So duplicated pair of homologous chromosomes are as such. Here are each sister chromatids. What will happen then, the diploid cell with duplicated chromosomes will enter into meiosis 1. Ultimately, in meiosis 1, homologous chromosomes are going to separate. Haploid cells with duplicated chromosomes are a result of that. And then in meiosis 2, the sister chromatids are going to separate. And you're going to have haploid cells with unduplicated chromosomes result. So the cell is going to divide twice. It results in 4 daughter cells, each with half as many chromosomes as the parental cell. In meiosis 1, the first division. Interphase, the chromosomes are replicated. In prophase 1, you're going to have homologous chromosomes pair up, and they pair up at their synapses. Ultimately, that pairing up creates a thing called a tetrad, where you have 4 sister chromatids in alignment. And you could see that tetrad formation right there as they line up at their synapses. At this point, a special event could occur that's going to increase the genetic variability of the resulting product. And that is called crossing over. And crossing over is going to be at a point called the chiasmata. So here you can see the chiasma in this particular picture here, transmission electron microscope picture of the homologous chromosomes paired. And right there you could see where crossing over would occur. Crossing over is going to take place on the insides of the homologous chromosome pairs. Ultimately then in metaphase 1, the tetrads are going to line up along the center of the cell. In anaphase 1, the pairs of homologous chromosomes are going to separate, and sister chromatids are still going to be attached by the centromere. In telophase 1 in cytokinesis, a haploid set of chromosomes is going to result in each new daughter cell. Each chromosome equals 2 sister chromatids. Some species, chromatin and nucleus, will reform. Then you have a stage in between prophase, or in between meiosis 1 and meiosis 2 called interkinesis. So here's a better division, or a better division. Here is a better illustration of what meiosis 1 looks like. So take a moment, pause the slide, and look through this. So after interkinesis, in which there is no replication of DNA, we go through meiosis 2, which is a second division, and that ultimately is going to result in the formation of gametes. So in prophase 2, there is no interphase, no crossing over will occur, the spindle fiber is going to reform, okay? In metaphase 2, chromosomes are going to line up. Anaphase 2, the sister chromatids are going to separate. Telophase 2, 4 haploid cells are going to result. The nuclei will reappear, and each daughter cell is genetically unique or different from one another. So they are genetically different or have genetic variability. So here, if you look, again, take a look through, look at this, examine each picture, look at what's going on, take a moment, pause. So events unique to meiosis 1 that are not in mitosis. Prophase 1, you have the synapses formation and crossing over at the chiasmata. In metaphase 1, the pairs of homologous chromosomes are going to line up on the metaphase plate. In anaphase 1, homologous chromosome pairs separate, but the sister chromatids are still attached at the centromere. So the source of genetic variation is ultimately in prophase 1 during crossing over. In crossing over, you're going to have exchange of genetic material. This is ultimately going to result in what we call recombinant chromosomes. You also have independent assortment of chromosomes, and this is a term that you learned in Mendelian genetics. You learned of the principle of dominance, the law of independent assortment, and the law of segregation. So independent assortment allows you to have assortment of chromosomes that will take place independent of one another. So all possible outcomes are able to occur. So you have this random formation of cells with all possible outcomes of genetic combinations. Random orientation of homologous pairs in metaphase 1. So sources of genetic variation also include random fertilization with the sperm. So any sperm plus any egg can result in genetic variation. So those are all sources of genetic variation there. So 8 million times 8 million equals 64 trillion combinations. Pretty fascinating. So let's look. Both are divisions of the cell nucleus, but let's take a closer look at comparing mitosis and meiosis and distinguishing differences. So both divisions are of the cell's nucleus. Mitosis is going to occur in somatic cells. Meiosis is going to be the formation of gametes. In mitosis, there is one division. In meiosis, you have two divisions. Mitosis is going to result in 2 diploid daughter cells. Meiosis is going to result in 4 haploid daughter cells. Mitosis is going to produce clones of the cell. Meiosis is going to produce genetically different, less than 1 in 8 million are alike cells. Mitosis is from zygote to death. In meiosis, it's females before birth follicles are formed, mature ova released beginning puberty. Purpose for mitosis is for use for growth and repair, and the purpose for meiosis is for reproduction. In mitosis, there's no synapses and crossing over. And here you can see a chart of all that, what we just discussed. So here's another good summary table to have in your book. So the role in mitosis in animal body enables multicellular adult to arise from a zygote, produces cells for growth, repair, and in some species, asexual reproduction. But in meiosis, it's production of gametes, which reduce the chromosome number by half and introduces genetic variability among the gametes. Remember that during, just kind of bringing it full swing then, the whole formation of our gametes is called gametogenesis. In the males, it's called spermatogenesis, and in females, it is called oogenesis. Ultimately, in males, all four resulting daughter cells are going to become spermatozoa. But in females, you get this unequal division of the cytoplasm, and therefore you get these three smaller polar bodies, which are going to disintegrate, and ultimately what you're left with then is one mature ovum that will be released. So that's it for meiosis and sexual life cycles. We will do a comparison of both this and mitosis in class as we go throughout the activities and labs. Thank you, everyone, and have a nice day.